It turns out that Western blotting is a way to see specific proteins and to apply the antibodies that we have talked about to SDS gel electrophoresis. So you can separate your protein by polyacrylamide gel electrophoresis, and you'll have the whole family of proteins that are in the mixture with your protein. And then you can transfer the proteins to a membrane. There are membranes that very tightly bind to proteins, and so you can wash the protein through and onto the membrane. And then you take the membrane with the proteins that have been transferred onto it, because you can't add antibody yet, because the antibody is a protein, and if you added it to the membrane right at this point where you've just done the separation and transferred it, then the antibody would bind everywhere on the membrane. But you can block the membrane with a generic protein. And what's usually used is non-fat dry milk. Has lots of protein in it, binds really well, and you just saturate the surface of the membrane with that protein. And then you have to wash it, of course. And then you expose it to the antibody. And after exposing to the antibody, you wash out the antibody again because you don't want it just lingering in random places. And then the sites where antibody binds, which is where your antigen is, are detected by a secondary antibody that has some sort of signaling system. And we'll talk more about those signaling systems in just a minute. So you can find on a polyacrylamide gel, using this Western blotting method, exactly where your specific protein is and in what amounts. And that can be applied in a variety of settings. You can also do it with 2D gels, where you run a pH gradient and then the SDS gel, and you transfer it to the nitrocellulose, blot the nitrocellulose with nonspecific protein, wash it off, bind your antibody, wash it off, and then use your detection system, and you can see a subset of this whole mixture of proteins can be identified. So if you want to know, for example, where in the cell is your protein, or what in a particular mixture of proteins is your protein there, this is a very powerful method and a very sensitive method for doing that. Now, I want to go back to the monoclonal antibodies and talk about sort of a similar use, but a different way of using the antibodies. And this is enzyme-linked immunosorbent assay, ELISA, and is often called ELISA. Some people will say ELISA, but I will be using ELISA. And this utilizes the fact that you can have a capture antibody that you attach to a support, and then you expose it to a solution that has the antigen that you want. Because the capture antibody links to the antigen, it too is immobilized on the solid support. And again, there are washing steps. You put your capture antibody down, you wash it off, you add your antigen, you rinse any excess off, and then you add your secondary antibody, and that secondary antibody has an indicator, and that's sometimes an enzyme that carries out a specific reaction that takes something from non-colored to colored, or non-fluorescent to fluorescent. It can sometimes be radio-labeled. There is some way that you can indicate the presence of the secondary antibody. And after you've exposed to the secondary antibody, you wash. And then you look to see where the indicator indicates that there is secondary antibody, which indicates where there's antigen, which indicates where that's bound to the capture antibody. You can also do it with a solid support, which is attached to the antigen instead of to an antibody. And you use the antibody to the antigen, and then you add again the secondary antibody with its indicator system and detect where viral antigen was found. And there are lots of variation of ELISA. These are the sort of the most common arrangements, but there are others. So again, this one has to do with immobilizing the antibody, having a target antigen and a secondary antibody. So now we have our antibody, and you can see its target. We have a secondary antibody that's attached to an enzyme that takes substrate to a product that we can see. And so we have a sample that contains our antigen. We attach the antibody to the solid phase. This is the one where the antibody is attached. And we bind to the antigen that's in solution, and we wash away everything else. So all we've got left is our antibody and our antigen. And now we use a secondary antibody that's attached to an enzyme to bind to the antigen. You can also have another antibody that's bound to an antibody. This can be sandwiched in many different ways. We wash all the excess antibody away, and now we add our substrate and look for our product, and we can read our results and see where was the antigen that we were interested in. And on the right you see controls for each of the rows, and on the left the results from different samples. And you can see by doing dilutions of the sample, you can get some sensitivity about how much of the antigen is present. And in the bottom row there isn't any. In the second row it's not so much. There's more in the first row and a lot more in the third row. And this can give you a measure of concentration. Sometimes it's just a yes or no. Can you detect it at this level or not? Other times it is you want to know the concentration depending on the particular application. So can you think of some applications for ELISA? Try to think of at least two. And which of those is the most useful application that you can imagine for this type of analysis? And can you think of different arrangements that might be possible for antigen and antibodies, or a series of antibodies and then of course something to indicate where ultimately the antigen, whatever you're targeting, is. It's a very interesting method, and it is very commonly used in a variety of applications.